So after we speak about what is meant by codon or triplets and how can we match every amino acid to its specific codon using the genetic code chart, let's speak about what is translation, where does it take place, and what are the steps of translation. So first of all, before we speak about where it happens and what is translation, let's look at the structure of tRNA. tRNA is the key player in translation. It's the molecule that is going to transfer amino acid into the ribosome. And if you look at it, this is a tRNA. It will have two binding sites, one at the top, one at the bottom. At the top, it will have a binding site for the amino acid. And at the bottom, it's going to have something called anticodon. Anticodon, it's going to be opposite to the codon, complementary to the codon, and it's going to bind on the amino acid. So, for example here, AUG, which is the start codon, the first codon in the amino acid as we explained before, the anticodon is going to be UAC, which is complementary to it. Opposite to A is U, opposite to U is A, opposite to G is C. So this is the structure of tRNA. And every tRNA can bind only to one amino acid. So what is translation in one sentence? How can we summarize it? Simply, it's translating the genetic code or the messenger RNA into an amino acid sequence. And it happens in the ribosomes. It means in the cytoplasm. It happens in all cells, since all cells have ribosomes, including prokaryotes, eukaryotes, plant cells, animal cells. And the end product of translation is polypeptide, which is a protein, or which is going to be the future protein. So what are the three steps of translation briefly? The first step is called initiation, then elongation, then termination, and they happen all in the ribosome. Now, the ribosome is made of two units, small and large subunit. Small at the bottom, large at the top. And it is made of ribosomal RNA as we explained before. But when the ribosome is not working, is not translating, the two subunits are separated from each other, are detached from each other. When translation starts, the messenger RNA is going to be attached to the small subunit, and AUG is going to be recognized, and this is the initiation of translation. It's the start codon, the start point. Always it starts with AUG. And what is this amino acid? This is MET or methionine. So methionine is going to be the first amino acid, and AUG is the first codon. And if you look here, the blue one here, UAC, is the anticodon, and this is the codon. So the tRNA is going to bind to the start codon, and it's going to bring with it the methionine. This is the initiation. After that, this is what the elongation, so, or how it proceeds. So after the binding of the first anticodon and the first amino acid, then the ribosome will be having its complete structure. The large subunit is going to bind, and the large subunit has three sites called E, P, and A. And remember, E always stands for exit site. So this is where the tRNA will go, and a new one will come carrying a new amino acid. So look at that. So there will be an amino acid sequence growing, and those are the tRNA. This process is going to continue. More amino acids are going to be added until there is a stop codon. So look here. This is 1, 2, 3 amino acids can bind at one time in a ribosome at one time. Okay? So this amino acid already put its transfer RNA already put its amino acid, and it's going to leave. A new one will come here bringing another amino acid based on the codon. So this is CGU. So what is the name of this amino acid? We have to go to the chart, look for CGU. The first one is C, second is G, U. So it's arginine, ARG. So this amino acid here is ARG, and so on. This will continue until termination happens. So when do termination happen? Termination happens when there is a stop codon in the messenger RNA. When a stop codon is reached, any of the three codons, AUG or UAA or UGA, what will happen? A release factor will stop translation and the amino acid sequence is going to be released. That's it. This is the free polypeptide or the amino acid sequence, which is going to become later the protein. And this marks the end of translation. After that, the messenger RNA is going to detach, the two ribosome subunits will separate from each other, and everything is going to separate. Okay, so this is the termination. Again, initiation starts with AUG, termination happens with one of the three stop codons, and then a polypeptide is produced. So this is how protein is made. We call this process protein synthesis or transcription and translation.